Check, check, 1, 2. Check, check, 1, 2. Check, check. 1, 2, 2, 2. Check, check. 1, 2. Check. 1, 2. Hey, test. 1, 2. Check, check, 1, 2. Test. 1, 2.
Testing. Testing, testing. Testing, testing. Okay.
Okay. Testing, testing. Okay. Is that all right?
Yeah, I hear
okay.
it.
Great. Oh, it's like a voice inside your head.
Now people can really hear me. Being with us, I can hear. Yeah, I'm loud and clear. I'm sorry? 10:05 sharp.
Wow, she's good.
Ready? Hello and welcome. Welcome to the Vitro Life and iGenomics Scientific Studio sessions. My name is Melanie Velasquez and I'm the marketing manager for iGenomics. Today I have the pleasure of presenting Professor David Gardner. He is the group director of ART, Scientific Innovation and Research for Virtus Health. Today's topic will be Age of Antioxidants. And with that said, I'm going to go ahead and pass it over to Professor Gardner.
Thank you.
You're very welcome.
Thank you. Well, it's a pleasure to be part of the Academy and to give this presentation on the age of antioxidants. So that's my disclosure slide. So, atmospheric oxygen, 20% oxygen, is probably one of the most detrimental things that the pre-implantation embryo in vitro could ever see. And I've been working on the embryo for 40 years now, and now we know for a fact that it affects gene expression, histone remodeling, the proteome metabolism, cleavage, and transfer outcome. So it's really not consistent with good IVF. Now, this review from 8 years ago shows that 3/4 of the world's IVF was done in the presence of 20% oxygen. Now, over the last 8 years, I'm sure many clinics have moved away from 20% oxygen, but we can't avoid it in the lab. We just simply can't avoid it. And that's because we see it during gamete collection, the preparation of eggs and sperm, during ICSI, FERT checks, grading, biopsy. And still some labs, I hate to say, do use 20% oxygen. And if you're one of those labs, please reconsider that. But we also have the generation of reactive oxygen species. We do that during the preparation of sperm itself. During cryopreservation, that actually induces reactive oxygen, and the metabolism of the embryo itself causes problems. It generates reactive oxygen species. And that can have negative impacts on our embryo quality. It produces fragmentation, damage to the membrane's DNA, and as you can see, there's a whole list there. So it's really something we need to avoid. So, enter antioxidants. Now, this is not a new concept. As you can see, the first paper on this topic was in 1991 by Noda, and he was looking at superoxide dismutase, trying to get mouse embryos through the two-cell block. And it was, in fact, that paper that caught my interest, and in 1992, we started on our journey looking at antioxidants. We know that individual antioxidants can have beneficial effects, but what we wanted to do is what happens when you add them as a group. So I'm going to show you a series of papers, and I want to acknowledge my colleague for the last 15 years, Mai Trung, who's really been the driving force behind all of these studies. So antioxidants in art. Well, we picked 3. Acetyl-carnitine. Carnitine because it's the most abundant antioxidant in seminal fluid, but it also has protective properties for the embryo. Alpha-lipoic acid, otherwise known as the antioxidant of all antioxidants. It has the capacity not only to be an antioxidant, but once another antioxidant's been reduced, it can, oxidized, it can reduce it back, so it keeps other antioxidants viable. And acetylcysteine, not only is it in itself a very powerful antioxidant, but it's also the precursor to glutathione, and that's one of the cell's major antioxidant pathways. So we looked at those individually, and they were all beneficial. So what we wanted to do was, what happens when we put them together? And what we found when we looked at pronucleate embryo culture, that whether embryos were grown in a group or individually, in other words, conventional lab systems versus that we would use in a time lapse, that when we used 20% oxygen, there was a significant benefit on blastocyst cell numbers when the antioxidants were present. We then repeated that at 5% oxygen and got exactly the same data. And so what's interesting is that even at 5% oxygen, the antioxidants are having a profound effect. Okay. Right, we then do embryo transfers, and as you can see, when you look at the control compared to the A3, the AOX group, you can see there was an increase in fetal weight. Now that is not an overweight like we worry about in human IVF, because actually it's still below the weight of an in vivo developed fetus. But what we are seeing is we're getting closer to in vivo. So we saw a 14% increase in fetal weight, we saw an increase in placental weight and crown rump length, and we saw a difference in implantation. So following that, we wanted to know what happens during IVF. So we went back using a mouse model, but this time did IVF. So in this case, we could do gamete collection with or without antioxidants. And what we showed, as you can see here, is that the control group in IVF had no antioxidants. And when we added antioxidants to just the IVF, we get a significant benefit. If we add the antioxidants just to the culture period, we get a significant benefit. But if you add antioxidants during IVF and embryo culture, you get the greatest benefit. So it's a bit like the amino acid story. you need them to be present throughout all of the procedures. We then went on to look at vitrification, because we were aware that cryopreservation induces a spike of reactive oxygen species. The driving force of this is as we're moving more and more to single embryo transfer, we're cryopreserving more blastocysts. So this is increasing. We're also through biopsy of PGT and a growing demand for oocyte freezing as well as sperm freezing. There are concerns that cryo induces downstream effects, so we wanted to investigate what antioxidants could do. And as you can see from this, again, what we did is we grew embryos in the mouse model to the blastocyst on day 4, then vitrified them, warmed them, and then grew them out to day 5 and analyzed their cells, or we did embryo transfers. And what we can see is that by the inclusion of the vitrification of the antioxidants in the vitrification solutions alone, we get a significant benefit. When they're present in the vitrification and warming solutions, we get a benefit. But when the antioxidants are present in the warming solution alone, you don't get a benefit. So that indicates that the real benefit of the antioxidants is during the vitrification process itself, which is what we would have predicted. Interestingly, we know that cryopreservation has an adverse effect on the epigenetic state. And in this case, we used histone acetylation of H3K27 and H3K9. And what we saw was that when we vitrified our mouse blastocysts, we saw a significant decrease in acetylation levels. However, when we included the antioxidants, we got an increase compared to the treatment group. Now, it was still less than the control, but at least we saw some amelioration of the effect. And again, we went on to do embryo transfers and showed that we saw an increase in fetal weights once more and crown rump length. So there was clearly a post-transfer effect by including the antioxidants during vitrification. And that occurred both at 5 and 20% oxygen. So where do we go from here? Well, what we wanted to do is actually then follow on and look at the downstream consequences of culture and vitrification with or without antioxidants. And this was really an epic study that Mai did. We recently published this at the start of the year in RBM Online. And we used RNA-seq to actually characterize gene expression in the fetuses and placentas to see any downstream programming effects. And what was shown was that here on the left, in the liver, there was downregulation of over 1,000 genes by cryopreservation alone and an upregulation of 900. And in the placenta, there was a few, 80 were downregulated and 131 were upregulated. But when we looked at the livers and placentas of fetuses that were derived from embryos that were cryopreserved in the presence of antioxidants, so that's the only difference. We cryopreserved them. So this is the whole thing, this one brief treatment. And we had this profound effect in the liver of the fetuses. But nothing really happened in the placentas. But what we can say from all of that is that vitrification, when you include the antioxidants, you can normalize to a certain degree the differentially expressed genes within the liver. So that's really very, very encouraging. And it turns out that these genes that are affected, several of them are involved in preeclampsia. So it has some clinical relevance. We've also done some clinical studies, pilot studies. This was a sibling oocyte study we did with colleagues in Japan, where only the oocytes saw the antioxidants, and then they were cultured in G1, G2 with antioxidants, so we didn't treat the sperm. And what we found when we did the embryo transfers, the control group is G1, G2, is that you can see the ongoing pregnancy rate per embryo transfer was 50% in patients under 35 and about 25% in patients 35 to 40. But when we looked at the data for those eggs and embryos that had seen antioxidants, we saw a similar pregnancy rate in the under 35s, but we didn't see the loss of pregnancies in the over 35s. And this has been followed up by Mitsumoto, who was one of the authors on the paper, but this time looking at both sperm and oocytes being exposed. So rather than just the oocytes, he actually treated the sperm with the antioxidants as well. And this was data presented at the ASRM, and you can see there was a slight difference in fertilization rates, almost reaching significance, but there was a difference in embryo quality, and a trend, again, to higher pregnancy rates in the older age groups. So again, it seems that the antioxidants are having a beneficial effect in the older age groups. Now, we've just completed a very large prospective randomized trial at Melbourne IVF, and we'll be able to share those data with you shortly. So, in summary, in all the mouse studies, we've shown that this combination of S-acetyl-carnitine, alpha-lipoic acid, and N-acetylcysteine together had a beneficial effect both in IVF and embryo culture, and improved embryo development not just at 20% oxygen, but at 5% as well, which really shows you that that oxidative stress is present throughout. The effects were best and more consistent when you have the antioxidants throughout the procedure, IVF and the culture itself. I didn't have time to show you the data, but actually we showed that the antioxidants led to an increase in intracellular glutathione and a decrease in intracellular hydrogen peroxide, which was really important. As a result of all of this, fetal development, both at 5 and 20%, was significantly improved after transfer in the presence of these antioxidants. We showed that antioxidants helped the embryo survive and go through vitrification, and it preserved their gene expression downstream in the fetus's liver. And in the human studies, the inclusion of antioxidants is associated with improved outcomes in patients of advanced maternal age. So it really is a very interesting and exciting time in this age of antioxidants. So thank you very much.
Thank you, Professor Cardinal. We appreciate your presentation. In a couple more minutes, we're going to have Dr. Carlos Simon presenting Endometrial Factors Matter. See you in a moment. Hello. Hello, guests. We welcome you. We're going to begin Dr. Carlos Simon's presentation in 1 minute. Please, we welcome you to attend the studio session. Hello and welcome again. My name is Melanie Velasquez and I'm the marketing manager for iGenomics. Welcome to our Vitro Life and iGenomics Scientific Studio session. At this time, I'm excited to present Dr. Carlos Simon. He is the president of the Carlos Simon Foundation. Today, he's going to be presenting Endometrial Factors Matters. With that said, I'd like to pass it over to Dr. Carlos Simon.
Thank you. Thank you. Can you hear me? Yep. Well, thank you for being here. And I really appreciate that you are interested in the endometrial factor. So what I'm going to do is just for the next 15 or 20 minutes, I'm going with you through this endometrial factor to fully understand that. First, I would like to disclose, this is not going. Okay, now. First, I would like to disclose that I am just working in my own foundation, and we are founding specific companies devoted to specific topics that come from our research, in addition to being the external scientific advisor of big pharma companies, including iGenomics. So what I would like to cover in the next minutes is basically to discuss about the concept of the endometrial factor, what precision medicine is, and to understand the two main topics within the endometrial factor, which has been basically the microbiome and also endometrial receptivity, to cover two basic issues that we have been going from the bench to the bedside for the benefit of patients. And this is what I would like to really inform you about. Regarding this, I think that this is something which is quite obvious, that nowadays we are transitioning from generalized medicine that is based on percentage population averages, to what is important, which is precision medicine or personalized medicine, which is based in your particular patient. We know for a long time that this is even more important to know the patient than the disease, because everybody reacts in a different way. So that's why precision medicine is being used for a long time now in oncology and make the difference between life and death. And this is the whole dream will be also to use it for reproductive medicine in order to improve our results. And somehow it is being used nowadays for stimulation, ovarian stimulation. We do not stimulate all patients in the same way, obviously. We do not treat our patients the same, but when the time comes, we treat all of them in order to transfer the embryos and in order how to treat the embryo. So this is just to gain knowledge and be able also to move this. And the place that we work is in the preconceptional space, because this is where the action is. Pregnancy starts here. If we take care of all of these factors, we will be better off, because once pregnancy is established, as a gynecologist, the only thing that we do is to be sure that the embryo grows correctly. If we find an abnormality, nothing can be done. We will do surgery later on, or the pregnancy should be ended. Or if we have a preeclampsia, we will just stop the pregnancy as soon as possible. But everything happens within this space. And this is what we have been working with, acknowledging the relevance of the embryo, of course. But in addition to the embryo, the mother has many things to say. And the first thing is the fact that once the embryo enters in the uterine cavity, day 4 or 5, what he's going to visualize is the endometrial microbiome, because there is a microbiome that exists in the luminal part of the uterus, and this is important to consider, and I will show you why. Then the embryo and the mother will interact and will cross-communicate. And in fact, what the molecule that the mother passed to the embryo, they will be very important, even modifying the transcriptomics of the embryo and changing things for the lifetime for it. And then after that comes the initial attachment, which is when the embryo will adhere to the mother, and this is the starting point of pregnancy, and later on the embryo will invade, and decidualization is key in order to allow the invasion process that will be shallow, and then we'll have preeclampsia, will be proper, everything will go okay. If it is excessive, we will end up having a percretism or acretism. And once the patient delivers, we will have an issue because the placenta will not detach. So all of this happens in this moment, and that's why we have devoted our work on that. So let's discuss about the microbiome. So we are not only surrounded by microbes, but we are living, microbes, they are living on us and helping us to do our functions. And we know that we have 10 times more bacteria than cells. And if you weigh about 70, 75 kilograms, 1 kilogram of your body weight will be because of the bugs that they are living on you, mainly in the digestive tract and in the skin. So this is something that we know. The issue is that until now, bacteria can only be cultured, and only 20 to 60% of them, they will be cultured. So that's why until very recently that we do not have next generation sequencing, all of our knowledge about bacteria was biased because of technical situation. Now with the advent of next generation sequencing is when we have been able to ask ourselves whether an endometrial microbiota exists. We know that the vaginal microbiota is not about that. We know that 10% of the microbes, they are in the urogenital tract, but this was a big question mark. What we knew about that is that this is the next generation sequencing works. All of these works they were doing by culturing the tip of the catheter when they introduced the embryo, and what they learned from all of this was that basically if they found those pathogens, this is going slow. Should I point to? Should I point to here? Yeah, sorry. So if you have, if those bacteria grow, basically we are getting a decrease in implantation rate and increased miscarriage rate. This is what it was known in the era pre-next generation sequencing. Then we worked since more than 10 years ago, and Inma Moreno was the main investigator of that. We start to trying to understand using NGS basically to analyze the bacterial DNA, and if we are able to analyze, this is the 16S ribosomal RNA, those hypervariable regions teach us what is the identity of the bacteria. So by doing this, we learn that indeed there is a microbiome that is present in the uterus, which is different from the one that is present in the vagina. Is close, but 20% of cases they are different. And well, I will pass this like that. And the thing is, in this very first pilot study, what Inma learned is that when you have what we call good bacteria, which is lactobacillus, the blue, to more than a certain percentage, 90%, we call lactobacillus dominant microbiome, results, as you can see, they were really good. However, the same set of patients with non-lactobacillus dominant microbiome, pregnancy went from 70 to 33%, miscarriage from 16 to 60%, live birth goes down. So this was an initial observation from a pilot study that teaches, okay, we have, it will be interesting to see the microbiological angle of the endometrial microbiome there. And this is what we did. So there is a number of published evidence since our work of 16, not only from us, but also from the group of Richard Scott, founding the existence of this endometrial microbiome. And even it was this paper coming in Nature Communication in 2017 that they found that there is not only an endometrial microbiome, but a microbiota continuum throughout the reproductive tract with different type of pathogens. You see the vagina, and then the uterus, the tubes, and even the peritoneum. So there is an ecosystem there that we have to know in order to learn what is normal and what is dysbiotic. That's the point of this. And we were fortunate, and we identified for the first time ever the first glimpse of the endometrial microbiome in a normal human pregnancy at the 4th week of pregnancy. This was a patient, this was my patient that I was fortunate to see. This was a young patient with very good embryos, euploid embryos. I transferred the embryos once. She became pregnant, but she got a spontaneous abortion. And this is the microbiome here in your right hand of the microbiome once the patient miscarried. Because of the miscarriage, we did a curettage, we gave them antibiotics, and she came back in September. She was 1 day delay of her cycle. So I just took endometrial fluid without knowing that she was pregnant, and I find out this 7 days later. So you have there what is in the endometrial microbiome of a normal human pregnancy with the embryo there. So we know that this is the proof of concept that this should be a good microbiome. As you see, everything is lactobacillus, mainly lactobacillus iners. So let me show you. This is the video that you can find in American Journal about this paper. Just 1 minute. Can you just... No? Yeah.
Around 9% of the bacteria in the human body can be found in the female reproductive system, where bacteria may facilitate or hamper human conception. Using 16S rRNA sequencing and functional metagenomics, we described the human endometrial microbiota in a patient who had an 8-week spontaneous clinical miscarriage with euploid embryos, and for the first time during a successful pregnancy in which the endometrial fluid was sampled at 4 weeks of gestation. Lactobacillus iners was the most prevalent microbe found in the endometrium during early pregnancy, associating its presence with defense mechanisms and basal functions. The microbial profile prior to the spontaneous abortion had higher bacterial diversity and lower lactobacillus abundance, showing an unstable functional pattern characterized by transposisis and insertion elements. We found taxonomic and functional differences between the microbiota found in the endometrial fluid collected during an early successful pregnancy and prior to a spontaneous abortion in the same patient. Further investigation of the microbial community in the female reproductive tract. Around 9.
And we continue with further investigations, and this paper was published January this year, by which we just did a multi-center observational study with about 300 patients in 8 different centers worldwide. And what we learned here, this is the flow chart of the study. This was, again, observational. We did not intervene. We just obtained the endometrium and the endometrial fluid, and then we follow the patient and to find out whether she had good outcome, which is live birth, or she has a bad reproductive outcome, which is clinical miscarriage or no pregnancy. So what we learned, you can find the paper. So this is the message. What we found is that when you have a poor outcome, this is the signature of the bacteria that they are increased, related to no pregnancy or just clinical miscarriage. And you can find this in endometrial fluid and so in the endometrial biopsy. So this is the signature of pathogens related with poor outcome when you analyze this. And on the other way around, this is the signature of the good guys that they are increased in live birth compared to bad outcomes. So as you can see, lactobacillus was almost nothing, below 95% as well, both in endometrial fluid and endometrial biopsy. So all of this collecting the idea that the endometrial microbiome exists, and there is a signature which is associated with reproductive outcome. And now studies, interventional studies, they are coming. I just read one from Japan, that they are just already treating the patient and then transferring the embryo afterwards to see at what extent we can improve the results. So in summary, from this part, the endometrial factor, considering this microbiological angle, the point that I want to make here is there is a pathogenic profile associated with reproductive failure based on this microbiome. Lactobacillus is consistently enriched always in endometrial fluid and endometrial biopsy. Both microbiota, they are not equal, endometrial fluid and endometrial biopsy, but they are very consistent because there are some pathogens that they are extracellular, this is the main difference. And of course, our point is that the endometrial microbiota should be considered as a possible emerging cause of implantation failure, and this is another angle for us to learn about that and to improve the quality of care of our patients. And the next step is endometrial receptivity, which is basically the point that I want to make now, and it's based on the ability of the embryo to adhere to the endometrial epithelium. And this is possible because the endometrial epithelium changes. It is transformed. It's what we call the plasma membrane transformation. If this doesn't happen, pregnancy will not occur. I say this many times, a human embryo can adhere to any surface, can adhere to the peritoneum, to the tube, you have an ectopic pregnancy, to the plastic, you have a transfer catheter that you have to try again, but will never adhere in a non-receptive endometrium. And most of the time, in your menstrual cycle, you are non-receptive. There is only a window of opportunity that this plasma membrane will occur, and now an embryo, which is an immunologically and genetically different organ from the mother, will be able to attach and to make it happen. And in fact, this is the reason for the continuation of the species. So we always have tried to understand how receptivity occurs from anatomical medicine to molecular medicine. As you know, anatomical medicine failed for many times. We have the Noyes criteria. And then from more than 20 years ago, we and other groups, we tried to use the new Google glasses of the transcriptomics to understand how can we identify objectively when the window of implantation will be ready by analyzing the transcriptome of the endometrium at that moment. And at that time, we use the whole tissue, the whole endometrium. And by doing so, we identify a transcriptomic signature that we later on verify using single-cell RNA-seq, and we published this in Nature Medicine. By doing the same, but now cell to cell, we analyze those cells, and we were able to identify that when we are now separating the different compartments, this is what happens in the epithelium. This is what happened exactly where the implantation, the addition occurs. So as you can see, with your bare eyes, there is a difference in color here. There is a step, and basically means that some genes are turned on and others are turned off immediately. And this is the reason why the window of implantation opens, and this set of genes coincides with the genes that we have in the ERA. And that's why the whole point to say, okay, now the window is open. This is the personalized window of implantation of your patient. This is completely different from the stroma. The stromal compartment, as you see, you don't have this abrupt change. You rather have a more discrete changes that go through the decidualization. And decidualization starts before the window of implantation opens. So we learn all of this from the basic part. But at the end of the day, the clinical lessons was there is a window of receptivity. And this window of receptivity is not for everybody, is not the same. The window is about 40 hours, but some patients need more time or less time for the progesterone to produce this. The window of implantation is directly related to the activation of the progesterone receptor. And progesterone receptors, they are different. You may have polymorphism. You may have different phenotypes of the progesterone receptor. And that's why the reason all the patients, they do not have the window of implantation at the same time. The majority, 70%, will be after 5 days, 120 hours of progesterone exposure. The majority, 70%. But in 30% of cases, patients will need more days, more exposure to progesterone or less exposure to progesterone. And this is the concept of individualizing the window of implantation and knowing when a patient will be ready to be transferred. And according to that, we create the concept of personalized embryo transfer, initially for our most difficult patients, those that fail everything. And the concept is very simple. Instead of transferring the embryo only when the embryo was ready, let's do it when the embryo is ready, but also the personalized window of implantation of the patient is there. And according to that, then we move forward, and this was our first paper showing this in patients with repeated implantation failure. And this has been the very last paper, also in repeated implantation failure from an independent group, again validating that those using ERA improve clinical pregnancy and implantation rate. But the best that I can offer you about the data that has been generated in the last 6 years or so, you can find in this paper, table 1, you have all the papers that they have been working on both recurrent implantation failure and good prognosis using clinically the ERA and see what happens. And also this is a meta-analysis published in April this year, taking into consideration, again, these two main outcomes that I would like to revise with you. So this is the meta-analysis, analyzing all the published data at that time, and they divided in patients with poor prognosis, those with recurrent implantation failure. Remember, recurrent implantation failure means patients that they have low chance of pregnancy. Typically, we identify them because they have failed 3 times, at least 3 times, good embryo transfer, and this is what we call poor implantation rate, and then patients with good prognosis. So for those with recurrent implantation failure, all the data that are there, the authors indicated that personalized embryo transfer guided by ERA significantly increases the chances of pregnancy for non-receptive patients. with recurrent implantation failure of endometrial origin. In a way, the point is if you use ERA, the clinical outcome will be similar to those with normal patients. So this is something that we were just trying to find out. However, for the good prognosis patients, the data that are there, you have live birth rate goes from 39% to 53% using ERA, but this is not statistically significant. So even if you have the difference, they say that for good prognosis patient, it's not clear at what extent we will help us to improve. But the thing is, for good prognosis, there are 2 randomized clinical studies done. One that is published, it was by our group, in which we found that per intention to treat, we found no differences because it was a high dropout of 50% of the patients for mechanical reasons. But when we analyzed per protocol, we find out that there was an increase in implantation rate, pregnancy rate, cumulative pregnancy rate, and cumulative live birth rate compared to frozen and fresh. For live birth, there was an increase of 13 points and 10 points versus frozen and fresh. However, this study was powered to find out 15% differences, so it was not significant. And the other study that has been sponsored by us, has been done by Shady Grove, is not published yet. They use embryos, PGTA analyzed embryos, and they show no clear results, no clear differences, but it's not published yet. But it was presented last year as an abstract in this meeting. Now, recently it comes this retrospective study from the center that I used to work in, and it was interesting for me to see that in the same center, with the same patient population, the same topic, 2 studies give us different results. And when we just saw what happens there, you can see this is the retrospective study left, and this is our randomized clinical trial, and those are the same patients in the same center. So, but the interesting thing was that when we learned the paper, the data in the retrospective study, they did not include those patients that it was involved in the randomized clinical trial with good results. And seeing the numbers of patients that they have been considering, 88, and the one that involved in our RCT, well, we don't know why they were excluded, but certainly this is a bias in addition to what a retrospective study is. So this is just to show you pros and cons and what has been published about that. The thing here is that in this meta-analysis, what we can see is that in recurrent implantation failure patients with these numbers, this is the pregnancy live birth rate that has been reported, and they are really, the conclusion is that they're really helping these patients. And for patients with good prognosis, as you see, this is the number, 39% with no ERA, 53% with ERA. But even so, as you can see, the percentage that we obtain in patients with implantation failure with ERA, they are similar to patients with good prognosis without ERA. So finally, I just want to show you this because it's something that we have always seen. Once you create something, there is always a panacea paradise. Later on comes the harmful hell that Dr. No is saying that's terrible. And finally, we will have a consolidation period, and the reality should come from the consolidation of studies and the proper analysis. And it happens for each test. Also happens for PGTA, and of course, ERA is no exception. And with that, I think that I just passed my time, and I just want to thank you. We are continue working on that to make that pathologies of affecting the uterus will no longer be an impossible barrier, and we are doing this in a foundation with the basic research team that are working on that. Well, thank you very much. If you have any questions, I will be here.
Thank you, Dr. Carlos Simon. Thank you, everyone, for joining us. As mentioned before, you can join us live on iGenomics YouTube, iGenomics LinkedIn, or VitroLife LinkedIn channels as well. Upcoming next, it'll be Dr. Beatriz Rodriguez She's a regional scientific advisor and account manager with Igenomics, and she's going to be presenting optimizing PGTA results and clinic data analysis. We'll be going live again at 1 p.m. Pacific time. Looking forward to seeing you then. Thank you for coming. Thank you.
test.
Sound well? Okay. Perfect. Thank you.
Hello and welcome back. Right at this time, we would like to present Dr. Beatriz Rodriguez. My name is Melanie Velasquez. Welcome to this scientific studio session. We're live here at ASRM 2022. Dr. Beatriz Rodriguez will be presenting optimizing PGTA results and clinic data analysis. With that said, I'd like to present Dr. Beatriz Rodriguez.
Hi, thank you very much, Mel, for the introduction, and also thank you everyone that is attending this live streaming session online. I think that for today's topic we have something exciting. We're going to have the chance to understand three key things. The first thing is, what is the data that you should be considering in the case that you want to know how are your PGT results going? The second thing is, where can you have access to that information? And the third thing, I am excited about this third thing because as someone that has been working inside of an IVF lab and that then moved to the industry and started working from the genetic testing perspective, is what is the connection between those rates and that data and the procedures and the protocol that you do in the lab. So the first thing is, what is the key information that we should be considering? That is what we refer to as key performance indicator. This is a publication from 2017. This is a consensus that was reached by ASRM and also scientists at Alpha. What they decided is that they put together a panel of 12 key parameter indications that were defined as those related to the core business of the ART laboratory. So within these 12 parameters, there's only one that was related with the PGT results, and that one is referred to as successful biopsy rate. That means what is the proportion of your embryos that you biopsy and that you load the cells in a tube and that you send to a PGT lab and that you get an informative result. Successful biopsy rate is also referred to as informativity rate. And that implies two things. When you have a result that is non-informative, it could be either because there's no DNA or it's not amplified, or there was DNA but the quality of the DNA is not enough. It might have degradation or it might be fragmented. So in that scenario, it would fall within the non-informative category. So how do you check that information? So now with this tool that we have at iGenomic, which is the clinical dashboard, you can have access to this online. So you have your data on demand. Within that dashboard that you can have access, if you're working with us and you're sending your PGT samples to iGenomics, you can request access to this and you can have all this information. So the way it looks like is that you have two main panels. There's one panel which is for the PGT-8 related results, and there's another panel which is for the endometrial test or ERA related results. Within the PGT-1, you have three main dashboards. The first dashboard is what we call the overall view clinic. And in this one, you can have multiple different information that is key for you to understand how your lab is performing. So the way it works is that within the horizontal, you're going to have all the parameters that you can check. So that be the percentage of normal or euploid embryos, the percentage of informativity, maternal average age, and many other things. That's going to be listed in the horizontal line. You can also filter each of these parameters, and that's going to be shown in the vertical. And those are the filters that you can apply to each of the parameters in the rates that you can check. The one in black is the one that we have just explained, which is the percentage of informative embryos. And that is very relevant for you to understand how the biopsies and the loading process is working in your lab. There's another key thing for these dashboards. Once you have selected the parameter and once you have selected the filters, you're going to have here in the middle side two columns. So this dashboard is going to make a comparison in terms of the timing. So for example, you can see what is the percentage of informative embryos in the current year versus the previous year. And you also have a breakdown per month. That is very interesting because in my experience doing this troubleshooting with the labs, we have been able to conclude or to at least have some hypotheses on problems related with non-informative rates just because of the month breakdown for this information. So sometimes it's related with a new protocol, new technician, new embryologist, and that's something that's very useful information right here. The second dashboard within the PGT panel is the clinic comparison. So as you can see, the horizontal and the vertical is the same parameters and the same filters. The difference is that in this case, you can compare the results from your lab and from other labs. So that's going to be other labs within your network. That's going to be on group 2. You're always going to be group 1. Another option is to compare your results with iGenomics USA. And the last option is iGenomics Global. We think that this is very important because this should always be a two-way street. It's not about how well you are performing the biopsies or the loading procedure. It's also about how well is iGenomics performing. Just to give you an idea, the benchmark that was published in this study from the Vienna Consensus is 95%. So ideally, you should be at least at 95% having informative results. What we have currently as average at iGenomics USA is 98%. So that's something that I think it makes us at iGenomics very happy, is that the quality of our results is very high, and you could be easily within that 98% of informativity rates. And I can tell you that I have clinics that are currently at 99%. That's almost unbeatable. It's almost 100% of the samples that you send are going to have a result. And the last dashboard is also very interesting because everything that is non-euploid or non-normal is going to be considered an abnormal result. But under that umbrella, we have multiple different options. You could have an embryo that is aneuploid or segmental aneuploid, or mosaic, chaotic, and then you can also have the information for each of the chromosomes. So all that information for that category of abnormal is going to be in the third dashboard. As well, within here, you can compare your results against another lab from your network, iGenomics USA or iGenomics Global. So let's imagine a situation in which you're below the benchmark, you maybe have 95 or 94% informativity rate, somewhere around there, and you want to boost your results. So what is that connection? That's what I was trying to explain. What is that connection between the procedure and these rates? Because once you have this knowledge, you have the tool to take your results to the next level. So let's review this. This is our loading protocol. Most of the people that have been working in an IVF lab or in the clinic in general, they would think that this is the most easier protocol. And in some ways it probably is, because at the end of the day what you're doing is, once you have the biopsy, you're going to collect those biopsy cells, the trophectoderm cells, you're going to wash it in our buffer, put it in the tube, freeze it, and send it back to us. So it's supposed to be easy, but I'm going to use a sentence that I learned here in the US, which is the devil is in the detail, and I think that absolutely applies for this protocol because there's some details that are going to have a big impact on your results. So for example, for the first step, one of the recommendations that we have in the protocol is, before you start with the biopsy and you have the embryos out, you know that the faster that you work is the better, not only for the embryos, but also for the biopsies. Why? Because if you have those cells after the biopsy in the hood, sitting there while you're labeling the tubes, preparing the kit and everything else, you have more chances for having DNA fragmentation, and that's going to be related with your non-informative results. So one of the things is just be prepared before you take out the embryos and start the biopsies, and have all of the tubes, everything ready for the loading procedure. The second thing that is crucial in this first step is to have intact cells. So if there was any kind of lysis during the biopsy because of the laser or the flicking method or anything else, that's something that we don't want within the tube, just because that could mess with the diagnosis, because it might have an effect in, for example, having an increased rate of mosaicism results. Within the second step, which would be the wash this, there's a few things that are very important. The first thing, when you are working with embryos, you want to have 37 Celsius degrees, just because you want to keep the embryos in a good environment. However, that's not the case after the biopsy. Once you have those cells, you just need to be working at room temperature. So just be sure that within your hood, it's an unheated surface. That's very important, and it will be related again with the quality of the DNA and the non-informative results. The second thing that is relevant is the buffer. So most of the times what happens is that we have clinics that are not working with a single genetic testing lab, and you might have buffers from multiple different laboratories. It's very important that you only use the buffer specific to that genetic testing lab. In our case is the washing buffer from iGenomics, so just be careful with that. It's very important and might have an impact in your informativity rates. And finally, it's the oil. So once you move to the washing dish, it's important that you're not using oil anymore. So what is the connection with using the oil and the informativity? Well, if there's oil within the tube and with the cells, once we put the buffer for the amplification, it might have an impact, negative impact in that amplification process. So it's very important that the cells are clean and that there's no oil in that dish. So the next step is the PCR collection tube. So there's two things that are very important. The first one is the volume. So we need to have a very concentrated sample. Take into account that you're only putting like 5 to 10 trophlectorium cells. It's very small amount of DNA, so it's key that you put a very small volume. So we recommend nothing more than 5 microliters, and it's important to take into account the micropipette that you're taking. So for example, if you're using a mouth pipette, it would be very difficult to measure the volume, so we recommend using something that you can really measure the volume, and be careful with this step. And the next thing is the location of the samples. So you can do dry loading or pre-loading, but what is key is that once you have the tube, the sample is going to be in the very bottom of the tube. And that's why we also recommend, that this is not mandatory, but it's a recommendation, that you include a spinning right after you load the cells in the tube. The reason is that, again, we want to ensure that the sample is going to be in the bottom of the tube. Why is this important, and how is this correlated with the non-informative results? Well, once we receive the samples, we're going to put the buffer for the DNA amplification, and if the buffer doesn't reach the cells because they were in the wall of the tube, it's not going to get amplified. So it's going to be results as a no DNA. So even if you loaded the sample, if they were in the wall but not in the bottom of the tube, that's not going to get amplified, not going to get sequenced, and it's going to be a no DNA. So it's very, very important. It's also very important that this happens before you freeze the samples, because otherwise, even if we spin the samples at our lab at iGenomics, they just get stuck to the wall and they're not going to go down. And then lastly is that you have to freeze the samples before shipping them to iGenomics. And this is also related with the location of the sample, because let's say that you actually did put the samples in the bottom of the tube, but the sample was not frozen, and once we received it, take into account that it's going to be moved, it's going to be in a box that is well protected, but at the same time, people is placing them and moving around with the box, so the cells might be misplaced. So it's very important that you freeze the sample so that we ensure that it's going to remain in the bottom of the tube. Also, that's going to have an effect in the DNA quality, because we want to avoid extreme temperatures, that meaning too hot temperatures, but also too cold temperatures. So it has to be frozen at minus 20 Celsius, but not minus 80. So what I was saying a little bit earlier is that even though the official definition for KPI is that are those related to the core business of the ART laboratory, I strongly believe, as someone that has been in both sides of the story, that this should be a two-way street and that we both have a responsibility in the results. So on our side, from iGenomics, one of the things that we do is that we have a whole scientific team that is here to help you troubleshoot your results. All of us except Alan are attending this meeting at ASRM 2022, so please don't hesitate to come introduce yourself. We are a team of scientific advisors, so you can also have direct access to the laboratory director at US and Canada, and also the diagnostic manager, and Juliana Cucci, who is our VP in science. So we're always here to help. This is our email in case that you want to request access to your dashboard, or in case that you want to troubleshoot your results, we can do that. And we also have training courses for biopsies and loading procedures, so don't hesitate to send an email and we will be happy to help. Thank you very much.
Thank you, Beatrice, for your presentation. We appreciate you taking the time to show us and helping us learn a little bit more about iGenomics.
Thank you.
Thank you. Thank you. With that said, our next presentation is going to be taking place at 2:50 p.m. Pacific time. It'll be Dr. Carmen Rubio. She is our VP of Research and Development at iGenomics. Her presentation will be an Embrace update and a new protocol for Embrace. Looking forward to seeing you then.
Check, 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 check. 1, 2, hey.
Can I practice? Hello and welcome back. Thank you all for joining us at today's Scientific Studio Sessions at iGenomics booth. We're here for the VitroLife and iGenomics Studio Sessions, and at this time I would like to present Dr. Carmen Rubio. She is the Vice President of Research and Development at iGenomics. My name is Melanie Velasquez. Today's topic is going to be the Embrace update with new protocols for Embrace. And with that said, I'd like to go ahead and pass it on over to Dr. Carmen Rubio. Welcome.
Good afternoon to everybody. Thank you for coming to see our latest updates about this non-invasive approach to preimplantation genetic testing of aneuploidies. We have been working in the last 6 years trying to find a non-invasive solution to test aneuploidies in the human embryos. This has been the dream since we started in the 90s with day 3 biopsies, analysis of just few chromosomes, then we evolved to the NGS, and now we have the technology ready to test a small amount of DNA in the culture media. So we can apply modified protocols of NGS to try to see if the cell-free DNA released in the culture media do really represent the chromosomal constitution of the embryos. We have developed the technology, we have done some studies that I will present in the next slide, and we have some knowledge about these events in pre-implantation development, but we have so many questions to be solved. We are still working on trying to understand the origin of the cell-free DNA, how we can improve the IVF protocols to minimize the risk of contamination, how we can improve the NGS protocols to have really very consistent results, even if we have a small amount of DNA. And for sure, we need to work in the clinical side. So we are conducting several clinical studies to assess the benefits of performing these technologies for different patient populations. So we started, as I mentioned, 5-6 years ago, and our first publication was in 2018 in Human Reproduction as a proof of concept that we were able to identify the presence of this cell-free DNA in the culture media. We understand the limitations of working with this sensitive biological material, and we learned that we had to implement some modifications in the IVF protocol and also in the NGS protocol. And with all these lessons learned from the previous publication, we conduct a pilot study that we published in 2019 in a small group of patients to understand how the concordance can be and the clinical impact if we select embryos according to this analysis of the medium. With all this in place, after the pilot study, we started the big multi-center study to see if the conditions that were working properly in an IVF lab can be extended to other different labs. So this is our second paper that we performed in collaboration with Genera Labs, with Laura Rienzi and Filippo Ubaldi. And I think that the main conclusion of this paper was related to the timing for media collection. So we were working with this lab. Embryos were not manipulated before collecting the media. They were PGTA patients, but they were doing the hatching of the zona pellucida right before the biopsy. So we were collecting the media before in embryos without any previous manipulation. So they were doing the PGTA according to the regular practice. Faster embryos, biopsy on day 5, media collection on day 5. Slower embryos, day 6 biopsy and media collection on day 6. We were checking the results and we learned how concordance rate were 63%, that they were double than in our first study. Informativity rates were so-so, 81%, but when we were testing the embryos on day 6, the concordance rate were 84%, and the informativity rate was very good. And I like to emphasize at this point that these were embryos without a previous biopsy, without a previous vitrification, just in regular physiological conditions. So with this, we started the multi-center study. The design of this study was for 2,600 embryos. So we published the interim analysis with 1,301 samples, in which in all of them we were able to analyze the trophectoderm biopsy and the culture media. And in a small subset of samples, we were able to collect the results of the inner cell mass biopsy. And I can tell that now we have finished this study. We have already collected all the samples. We are analyzing the data. We are having more than 300 samples in which we have also the inner cell mass biopsy. And as you can see here, we are very happy to see that the concordance rate of the medium with the inner cell mass is as high as 86% and is kind of very similar to the concordance rate of the trophectoderm biopsy with the inner cell mass, that is just 89.6%. So these are our more recent results submitted to this year, and hopefully by the beginning of next year we will be able to publish the final results of this study. What I presented so far were the results with fresh samples, but we can apply this technology also to frozen embryos. We had a lot of centers asking us what to do if we have previously vitrified blastocysts. How long should we keep the embryos in culture after thawing to have enough DNA for the analysis? With this study that we performed in collaboration with Boston IVF, Dennis Zakas, and with ORCID in Dubai, we were able to identify that if we have day 5 frozen blastocysts, the best moment to collect the media is after overnight culture. So with day 5 blastocysts, overnight culture, and then you can collect the media in the morning and you can have good results. With day 6 blastocysts, we don't want you to culture one extra day, and if you culture only for 6-8 hours, is enough. We have enough DNA. So the idea for day 6 blastocysts is to thaw in the morning, and then early in the afternoon you can collect the media. With this, you can have informativity rates in the medium similar to the trophectoderm biopsies, and the concordance rates are very good for frozen blastocysts. For frozen blastocysts, as in previous publications, your concordance rates can be higher, at least 90%. So this is the protocol we have now for fresh embryos and for frozen embryos, and as you can see, we have very good concordance rate. I have to say that we never are going to have 100% concordance rate, in the same way that we are never going to have 100% concordance rate between the trophectoderm biopsy and the inner cell mass. So we are aware that we have mosaicism that can be something between 6 to 8%, in the same way that we have this in the trophectoderm biopsies. But we can control for the technical parts. We cannot control for mosaicism because this is a biological phenomenon, but we can control for the external factors that make this little difference in the concordance rate. So we need to control for contamination, and we have two sources of contamination in the culture media. The most important one, cumulus cells, maternal contamination from the cumulus cells that stay close to the embryo. And this is not a unique problem for the medium. This also happened with the trophectoderm biopsies. We had a session this morning talking about error rates in PGTA, and when we have a discrepancy in the gender, most of the cases is because we also have contamination in the trophectoderm biopsy. So we have to be aware that if we have surrounding cells close to the zona pellucida, we can have a misdiagnosis either in trophectoderm biopsies and also in the medium. So this is something we can control for, and we can minimize the risk, and also we can minimize the risk of external contamination from the surfaces, from the embryologies, with very easy tips. This is not like science fiction. It's very easy just to control for these sources of contamination. For the maternal cell contamination, we have implemented in the IVF protocol 2 series of washes, and it's very important. The most important step is day 4. So if in day 4 you have the embryos, if you still see some remaining cumulus cells, it's very easy just aspirating very smoothly to remove these cells. On day 4, they are not so attached to the zona. And then to move the embryos to a fresh drop of media, and it's working very nicely. And then you really minimize the risk of contamination. For external contamination, very basic aspects. So if you are familiar working with PGTM for monogenic diseases, it's the same policy to try to avoid contamination, covering properly with a cap, with the mask, with the surgical gown. Very important to cover the arms, and also to clean very well the hood and all the material that you are going to work with during the process. So this is how we work. This is not perfect. We know that we need to wait until day 6 to have enough DNA to have a reliable result. And the big question from the embryologists has been always, is it detrimental to extend the culture of the embryo to day 6? I can say that we are gaining more and more evidences that in a optimal, in a proper culture media system, this should not be a problem. And this is the study published by the group of Denis Sakas. They sent to ASRM last year, and they have observed that using our protocol, the live birth rate is similar to in cases of PGTA in which embryo were biopsy on day 5 or day 6. So extending the culture to day 6 in all the patients have similar live birth rate than doing some of them on day 5 or on day 6. So we are gaining more evidence that we can really work with this protocol with the aim of improving clinical outcomes, but still this is not perfect, as I mentioned, and we are proposing to use the information of the cell-free DNA as a biomarker, to add the information to other systems you had in your lab, to incorporate this information to morphokinetics, to morphology, to better score the embryo. So we establish a ranking for the embryos according to the probability of each embryo, or maybe of each media, to correspond to a euploid blastocyst. We have the big experience of the multi-center study with more than 1,000 samples, so we have the results of the media, we have the results of the trophectomy biopsies, and we have established a system in which we can rank the embryos according to this probability. So this is the way in which we are proposing the application of this technology in the clinics. We are starting little by little. We always say it's very important to start establishing a very close collaboration with all of you in the IVF labs. We do a validation of each lab, and we do a very close follow-up to understand how can we adapt the protocol to make it more user-friendly in your facilities. With this, I'm based in Spain, in Europe, and we have done so far 41 validations, so validation for 41 centers. Our average concordance rate is 86.4, and I'm very glad to say that the last 4 centers that we have validated in August and September, they had very good concordance rate, and we have very good concordance rate with EmbryoScope too. With EmbryoScope, we work with a little higher volume, but it's working very nicely, and in the last 3 cases that we have validated, combining EmbryoScope with Embrace, we achieve 100% concordance rate. So now we have a protocol for fresh embryos, for frozen embryos, for conventional incubators, and for EmbryoScope. So if you are happy with the idea, just contact us, because we can try to adapt the protocol according to the design of your lab. And what can be the benefits regarding the clinical outcomes? We have a glimpse of the potential benefits that from our second study. In this study, the embryo transfers were performed always according to the results of the trophectomy biopsy, and in a blinded way, we were analyzing the media. So embryos were transferred according to the biopsy, and then the IVF center shared with us the clinical outcome, and then we unblind the results of the media, and we learned that even though the trophectomy biopsy can be euploid, in some cases we have the aneuploid media, and then in these cases we observe a high miscarriage rate and a lower implantation rate. So this was showing us that maybe the media is giving important information for the patients. And in this last slide, I'm presenting you the data from one of the IVF centers that is collaborating with us in Spain, is Ruber International. They were comparing the results in patients doing non-invasive PGTA and morphology. They presented this last year at the ASRM meeting, and you can see how non-invasive PGTA in pink behaves very similarly to the PGTA according to ongoing pregnancy rates, and how the ongoing pregnancy rates were more than 10 points higher than with normal morphology. So these were the results of last year, and this year they presented update results at ERRE, and you can see how ongoing pregnancy rates with non-invasive PGTA can be as high as 75% compared to 55% with morphology. So this is the idea, that you can start little by little in your center, and you will understand in your patients where to apply this technology. In this particular center, they are applying the technique in good prognosis patients, patients below 38 years of age, or ovum donation, because they are doing PGTA in the patients of above 38. So this is the end of the talk, and the idea of this, as I mentioned, the conclusions is that we have learned that there is embryonic cell-free DNA in the sperm blastocyst media, that this can be a powerful biomarker to diagnose or to understand the presence of aneuploidies in the embryos. We are giving the approach of using prioritization, and in this scenario, we can have an attractive option for patients to increase the clinical outcomes in general for any population. Thank you very much.
Yeah, absolutely, no problem. Thank you all for joining us. Our next presentation is going to be at 3:15 p.m. It'll be Pacific time. Our presenter is Deirdre Xander Fox. He is an associate professor and a scientific director at Monash IVF Group. His presentation will be Incorporating Genomics in an IVF Program. Looking forward to seeing you all then. Hello and welcome back. Thank you for attending the VitroLife and iGenomics Scientific Studio sessions. At this time, we're going to have Associate Professor Deidre Zander Fox. She is a Scientific Director at Monash IVF Group. Today, she will be presenting Incorporating Genomics in an IVF Program. That said, I'm going to go ahead and pass it on to the professor.
Thank you. Thanks so much. All right, just starting with a brief introduction. As was said, I'm also Chief Scientific Officer for Monash IVF Group, as well as being Regional Scientific Director. And I also have some university positions as well at Monash University and also University of Adelaide. Just to start with, just to give a little bit of background from where I'm from Monash IVF Group. We're a very large IVF group that's based in Australia and also Southeast Asia. We have 24 clinics within Australia, and we also have 5 clinics that are based in Southeast Asia. And we are in all of the states and territories of Australia. We have 3 day hospitals, but also what's relevant to this talk is that we're fortunate enough to have 2 genetic laboratories within our organisation. So we're able to perform all of our genetic testing within house. We also have large ultrasound practices as well, which ties into a little bit of our genetic testing offering as well. So this is inside our genetic laboratories within Monash IVF. This is our laboratory that's based inside of Adelaide, which offers PGT-A, ASRM and also NIPT testing. And then in Melbourne we have another laboratory which also offers PGT-A, SR and M. So our model of care for genetic testing spans preconception and also prenatal testing as well. Any patient that is looking to conceive a child, we believe should be offered carrier screening, and just depending on the results of that screening and whether or not they've had trouble conceiving in the past, they will then come into the IVF program where we can then offer them varying sorts of genetic testing as part of the IVF treatment cycle. So if our patients have had a high-risk carrier screening result, they can then access PGT-M. If they have a known structural rearrangement, such as a translocation, they can then access PGT-SR. And then if they may benefit from PGT-A because they have advanced maternal age or maybe recurrent miscarriage, then they can also access PGT-A. We also then have the ability for them to have non-invasive prenatal testing, if they are pregnant from 10 weeks of gestation as well. So we really like this model of offering because it does cover that sort of full spectrum of genetic testing throughout the early stages of conception. We think that it is really important to have genetic testing as part of your IVF offering because it does help our patients determine whether or not an embryo that's been created is likely to result in implantation and will hopefully increase success rate and decrease time to pregnancy when it is used appropriately. We don't use PGT as first line for all of our patients, it's certainly used where it's clinically appropriate. It also as well for our patients that do carry a known genetic disorder or translocation can help increase the likelihood of having a child born without genetic disease. And we also, like I said, we can extend the genetic testing to preconception and also early pregnancy as well. It does require, though, access to genetic counsellors within our teams. We have access to clinical geneticists and also a genetic pathologist as well. And we really do need excellent coordination between our clinical teams, embryology, the genetic scientists, to try and achieve the best outcomes for our patients. So what I'm just going to do now is take you a little bit through a little bit of our history of, we've been doing genetic testing for quite some time within our clinics. Like I said, we're quite a large clinic group, so we are really fortunate to be able to offer it in-house. But I think some of this also does apply that if you even can't have your own genetic laboratories, how you can perform the biopsy in-house and then send it out to an external lab. We've been doing PGT-A and SR since the 90s. It was introduced in 99. We were doing FISH for sex selection and then aneuploidy detection in 95. Brought in translocation testing in 2000. And then we moved over to PGTA using array CGH in 2013 using Blue Gnome 24 Shore. I'm sure everyone might remember that. And then we moved on to next generation sequencing in 2015. And our reason for doing this was it was a faster turnaround time, a higher resolution, reduced noise, and to do that we use VeriSeq. And then we are looking to transition to embryo map in very early 2023 at the latest. Being able to offer it in-house within our own clinics and control our own genetic laboratories has really benefited. We have been able to increase the turnaround time or speed that we can get the results back to patients. It's been able to decrease the cost to the patient and also gives access. We've been able to bring in-house genetic counselling support as well, which is very beneficial. And our clinicians do really appreciate having direct access to the genetic scientists that run the test and be able to provide discussion and advice for their patients. We also have been doing PGTM for single gene disorders for quite some time. It was brought in back in 1999, and then in 2015 we moved over to carrier mapping technology for many reasons, including the fact that there isn't that need for a customised test for each patient. You can use that one platform for basically everyone. From a genomics in IVF perspective, and the fact that we offer expanded carrier screening, it is really received well from patients to then be able to offer them PGTM within the clinical setting as well. It gives all our patients and doctors a very well-defined treatment pathway, and it also does with that PGTM service with the carrier screening, it is increasing the utilisation of PGTM and then allows us to turn the results around for patients faster. Also, we're very lucky in Australia because last year we had PGTM added on to our Medicare benefit scheme. So that means patients get funding from the government to be able to undertake PGTM if they're carrying genetic disorders or they have a translocation, which is fantastic. In addition, in Australia, in 2018, around $500 million was provided by the federal government to try and increase genomics research, including $20 million, which was for Mackenzie's mission. So Mackenzie's mission is a research project screening 10,000 couples within Australia, looking at a panel of 750 diseases. And if they come up as being at high risk of having a genetic disorder in their children, they can then access a no out-of-pocket Medicare cycle. This was initiated by patients who did a lot of campaigning and lobbying because they tragically lost their little baby Mackenzie at 7 months of age because they were both carriers of SMA or spinal muscular atrophy. So they have really advocated for change to our guidelines. That carrier screening should be routinely offered to all couples prior to conceiving. So this is demonstrating, I think, the importance of this testing to any couple who is considering having a baby, regardless of whether or not they're coming through an IVF program or not. So I just wanted to talk now a little bit about practical aspects to bringing genomics in-house if you don't have a genomics program in-house or you don't offer any form of biopsy. I think this applies to whether or not, like I said, you create your own genetics laboratory or whether or not you just want to bring biopsy in-house and then send the testing off to an external laboratory. Prior to starting a PGT process, all couples really need to undergo genetic counselling so that they know whether PGT is the right option for them. We have a few different ways of doing this within our group of clinics. The treating fertility specialist can do that counselling for PGTA. Also, because of the high uptake of this, we also in some clinics offer it via, they can watch a webinar and then have the consenting done after that. We also do have genetic counselling and clinical geneticists as well. And this is for PGT, SR and M. And in that they'll discuss the PGT procedure, limitations of the testing, and review the expected results and the accuracy. It's really important, we think, to emphasise that this is a screening test, it's not a diagnostic, so there is it is not 100% accurate and patients should know that. They will discuss any out-of-pocket costs to the patient and answer any questions. If they're undertaking PGTM, we also send a letter back to the patient that outlines the genetic counselling appointment, so they know everything that was discussed and they've got a record of that. And we also provide extensive information sheets as well to the patients. All patients need to sign, we have them sign a PGT consent form relevant to the testing type, clearly outlining all of the patient details, the tests that will be performed on their embryos, and like I said, a lot of limitations, that not all embryos might be suitable for biopsy, that embryos can be damaged, although rare, during the biopsy process because it is an invasive procedure. We might not get results for all the embryos, and again, as I said, that results are not 100% accurate. In regards to the laboratory side of things, there's some excellent references in how to set up PGT within a laboratory side and the embryology side. That the laboratory should be organised according to the guidelines for good practice in IVF, and there are excellent references on this from ASRM. You need a single embryo culture system. It needs to be clearly labelled so that you can trace and track every embryo. You need very strict witnessing protocols to minimise the chance of human error. And embryo identity and biopsy sample identity needs to be checked throughout the whole procedure so that that testing result can be unequivocally linked back to that specific embryo. The dishes, embryo biopsy dishes, need to be made up before the procedure and clearly labelled. And you need the biopsy drops to be spaced apart to make sure you don't have any merging. Consideration should always be given as well to one embryo per biopsy dish, and we do this for PGT, SR and M cases. And the laboratory should also have strict guidelines about what quality embryos you're going to biopsy, and also trying to be careful to minimise the number of cells that you take so you don't compromise embryo viability. In regards to if you do decide or you have access to a genetic laboratory within your facility, this needs to be maintained to the highest standard with very good quality control procedures that surround it. We have a very stringent quality manual. It is a huge document. And we also in Australia need formal accreditation for our genetic testing laboratories, just like it happens in America. All of our scientists are trained extensively in PGT, which includes a lot of theoretical training and also hands-on training as well. And it's all done by very highly supervised scientific staff. And the leaders of our labs also have a doctorate in genetics and 10 years experience in the field. I've got a lot about quality control within a genetics laboratory. I won't go through all of it in great detail, but really you need to make sure that you run your laboratory incredibly stringently. The dates of your reagents, the quality of your equipment, everything needs to be really well maintained to make sure that the results that you provide to your patients are excellent. We do extensive auditing of every part of our program constantly, and we also audit our data as well, the transcription that we do, the entry of that onto our databases, the generation of reports, and even file storage and backup to make sure that everything is going and is as high quality as possible, given the importance of the screening testing that we're doing to the patients that we're treating. We also participate in external QA programs such as Gen QA, and that allows us to benchmark our results externally as well, and extensive backup and contingency plans as well for the laboratory, making sure you have generator, uninterrupted power sources, also that the outcomes, if anything should happen, you have a power interruption, that you don't disrupt the genetic testing process. And we also follow up all of our patients' outcomes as well, make sure implantation rate is acceptable, clinical pregnancy rate, loss rates, and also the babies born, making sure that you don't have a high false negative rate as well. So just in regards to risk minimisation, this is something we focus a lot in and should be focused on very heavily if you are bringing genetic testing in-house and trying to make sure that you don't have high levels of risk in your program. These are just some examples of possible causes of misdiagnosis and the actions that you can take to minimise them. Contamination is obviously a concern. You need to make sure all of your consumables are clean. That you use ICSI for PGT, ASRM. Make sure that you have a clear labelling system so you don't transfer inadvertently an embryo that has come back as high risk. Having highly trained scientists independently review the results so that you have two different people look at it to make that final decision and recommend confirmatory prenatal diagnosis, NIPT, etc. for if you have a pregnancy. And also natural conception is a big one as well. If patients are going through PGTM, they really, especially PGTM, need to abstain from intercourse so that they don't have a natural conception when you're trying to ensure that they have a baby that's not affected with the genetic disorder. So we think genetic testing is a very important part of an IVF program. It's, a detection of couples at risk of a monogenic disorder. Upstream, before conception, we use carrier screening. Decreased time to pregnancy in our patients of primarily advanced maternal age or repeat implantation failure, we offer PGT-A. For patients who have a chromosome rearrangement, we offer PGT-SR, and then PGT-M if patients are at risk of giving a monogenic disorder to their patients and to their offspring, and then prenatal testing as well. Introduction of an in-house program, if you are lucky enough to have that capability, can be really beneficial. Fast turnaround time for results for your patients, you can control that. You then have in-house genetic expertise, access to genetic scientists, counsellors and pathologists. Our doctors really like having access to that on a daily basis, and we're also able to give a lot of personalised genetic care to our patients as well. But that lab, if you do bring it in-house, has to be very highly run with very high-level scientific expertise, QC, QA and risk mitigation as well. So I'll just finish it there with the references that are really, really important if you're considering setting up a genetic laboratory in-house. And thanks everyone for your time.
Thank you for being with us. Thank you everyone for joining us. Go right ahead. Thank you everybody for joining us. That will end our sessions for the day. That'll conclude those. We ask that you join us tomorrow for the iGenomics in Vitro Life Studio sessions. Tomorrow we begin at 10:15 a.m. and that is Pacific time. The presenter will be Dr. Tiffany Stankiewicz. She will be presenting Optimizing Endometrial Results. We look forward to seeing you tomorrow at ASRM 2022. Thank you.